Good morning and welcome to the week 3 re-record, the brand new edition with the newly formatted lectures. So today's a snow day, so I'm just doing this on my own. I don't have any students to bounce off of, unfortunately, but we're going to make this work. So we're doing, again, a little bit of out of order compared to the old style, but that made a lot of sense last week when we had everything related to blood all in one place. So the content we shoved forward was this inflammation and immunity content, as well as autoimmunity. And you can't talk about autoimmunity without talking about HIV. So that's the stuff that got kind of shoved out of order in comparison to the old style. The heart cardiovascular content was otherwise already here. So alterations in cardiac function, and we'll also cover heart failures and dysrhythmias today. All right, and I think we've covered this concept week 1. Transmission requires multiple events in order for one person to catch disease that another person or place was carrying as a vector. So in order for transmission to occur, there needs to be a reservoir, a portal of exit, a mode of transmission, a portal of entry, and a susceptible victim. The good thing about this is that we can literally break this chain at any point. And you could pretty much put hand washing at any one of these points. But that could be more complex than that. A reservoir could be human, animal, insect, or soil. So, for example, in parts of Brazil, cholera was being spread through water sources, so that water source would have been a reservoir. Or in another case, hand towels that were shared among people of a population. So eliminating the water quality issues or eliminating shared hand towels would have eliminated the reservoir in that case. This should be a lot of review content here. You have an innate immune response, which occurs on initial exposure to the antigen, and some of it's just physical, like your skin, you could consider an innate immune response because it's a physical barrier between you and the world. And the only way to break that down would be to cut into the skin. Your skin also has certain cells that are going to release lytic enzymes as well that'll help break down anything you're exposed to. Your specific immune response is slower to develop. It only works the second time you're exposed to a specific pathogen. And again, as review content, you should be able to remember and tell me about all of the cells involved in specific immune responses, all of the T cells, all of the B cells, and the cascade that occurs there. Immunization is incredibly important because it helps us develop specific immunity without having actually had the disease before. We develop specific immunity when we're exposed to the antigens of a specific disease, and then we can confer specific immunity. That cellular pathway of specific immunity will be activated on first exposure because the immunization exposed it to you the first time without you expressing the disease. Now, again, we do need a vulnerable host in our transmission. So if somebody's immune system is weak or their gut flora is vulnerable, or even if they're in a state of extreme emotional distress and there's stress in their life, that makes opportunistic infections more likely. And we'll see that quite a bit with our immunodeficiencies, including but not limited to HIV. There will be many, many cases in which somebody could be immunodeficient aside from simply HIV. So let's look at the mechanisms of inflammation. Inflammation is going to be seen in very, very, very many disease states. So this is just the physical mechanisms of inflammation. It has a purpose. Inflammation is something your body does for a reason. It's going to help bring immune cells to the site of injury, the site of cell death. It's going to help also bring nutrients to that site of damage. So it's there for a reason. It's going to help repair the tissue. It's going to help kill the invader. But it's also going to, in some cases, be physically harmful to us because it can go too far. So when there's an inflammatory event, first of all, there's a tissue injury. Maybe there's a physical problem, or maybe a chemical agent or a pathogenic organism. And the physiological response to that is that first the capillary bed is going to widen. There's going to be vasodilation. Now I'm going to remind you, hemostasis, last week we had vasoconstriction as the first step. That's the opposite situation. Hemostasis, blood clotting, requires vasoconstriction. Inflammation initially requires vasodilation, and that's going to help increase blood flow to an area. It's also going to help increase capillary permeability. You'll see an increase in the space between the capillary endothelial cells. This is, remember, is a simple squamous epithelium. And that widening of those spaces is going to help release fluid, so that plasma is going to shift into the interstitium during an inflammation reaction. We're going to have migration of white blood cells, so leukocytosis. We're also going to see in just a minute the concept of pavementing and chemotaxis, and that means the movement of cells as they're attracted to chemicals. And pavementing is the process by which these cells, they were rolling along through the bloodstream, but they're physically being pulled actually out into the interstitium. That's what we mean by migration. And we'll get to those vocab words in just a minute. We'll also have fever systemically. We're going to try to kill the microorganism by destroying it with heat. Of course, that can be difficult for us as well. And proliferation of white blood cells. There will be more white blood cells. And collectively, these things lead to heat, redness, tenderness, swelling, and pain. So again, this is where, yes, your body has a purpose for creating inflammation, but inflammation can also hurt you. If this happens across a joint, for example, you may lose your range of motion. There may be formation of scar tissue. There can be, again, excessive amounts of pain, more so than is necessary to get the benefits of this process. In severe conditions of inflammation, you can lose function. I used the joint as an example, but that could happen in different places. Let's talk about, let's see, I've covered most of this. Again, there's a little bit more vocab for you here. Emigration of leukocytes, margination, pavementing is a vocab word we'll go over. Chemokines, let's go over this word chemokine and chemotaxis and diapedesis real quick. So you may remember from AMP2 cytokines. Now, take a second to think about what you remember about cytokines. Usually I would bounce off students at this time and say, okay, what were the cytokines? Where did they come from? And the idea is we're going to get to the word T helper cells. T helper cells release cytokines. And we made this in kind of a story. Turn this into a story where the T helper lymphocyte recognizes the foreign antigen and in response releases cytokines. So cytokines are chemical signals, and the story we told is it's like they're saying, hey, you guys. A cytokine is a chemical signal that invites other cells around, like it's yelling at them to show up, get over here, and attack this pathogen with me. So a cytokine is going to invite cytotoxic T lymphocytes and memory B lymphocytes to the site of a pathogen, some kind of pathogen invader. And of course, that's going to cause those cells to do their jobs. So cytokine, you can also sort of call it a chemokine. It's just, again, this general term for a chemical signal. Things that are on a pathway that invites more immune cells to the party. Chemotaxis, where's my chemotaxis? I know it's on here somewhere. Chemotaxis means we're moving towards the chemicals. So remember, the cytokine was the hey, you guys. Chemotaxis is what the other white blood cells do when they receive that chemical signal. They're going to go towards that signal. So like it's taxiing towards the chemical, if you want to think of it that way. Signs and symptoms of inflammation, the 5 cardinal signs: that heat, the redness, the swelling, the pain. There's going to be acute inflammatory conditions and also chronic inflammatory conditions, right? So you can have, ow, I bumped my elbow, you'll have acute inflammation and it will resolve. Or it could be, I've got recurrent ear infections. It's happening over and over and over again. And these things keep coming back and they stick around for a very prolonged amount of time. And when you have chronic inflammation, it brings new problems along. Things like scar tissue development, for example. Neutrophilia means abundant neutrophils. So even though neutrophils are the most abundant white blood cell already, this is still far too many of them in a blood smear. Okay, erythrocyte sedimentation rate would be a laboratory test that could test for, among other things, inflammation. So normally, remember your blood and your hematocrit. In your blood sample, you have a certain number of formed elements, red blood cells, white blood cells, and platelets, versus the plasma, which is the fluid portion, right? Now, in a normal blood cell that has not been centrifuged, the heavy elements, the formed elements, will eventually float to the bottom. And that drifting to the bottom, that separation into heavier and lighter weight things, that's called sedimentation. Sedimentation is actually a general biology term. So the dirt, the grit on the bottom of a lake, that's called sediment, right? So you can sort of slide your hand along the bottom of a lake and that sediment comes up and the lake gets cloudy for a bit, but slowly it drifts back to the bottom and that's sedimentation, right? So same thing going on here. The red blood cells are going to drift towards the bottom of a blood sample very, very slowly. But during inflammation reactions, that actually happens faster. So these things actually become heavier. The formed elements become heavier and more dense compared to the plasma. Sedimentation actually does occur faster. There was actually some confusion about this in previous quarters, so I did look this up just to clarify that yes, we are expecting faster than normal sedimentation rate with inflammation. Now, again, inflammation is going to be any type of cell injury, exogenous or endogenous. It could be something from the outside, maybe we got punched, maybe there was bacterium, or maybe something endogenous happened within our bodies, maybe autoimmunity, for example. And then, of course, any condition associated with inflammation and an itis. That's our basic medical terminology. We talked about acute versus chronic. The time scale for acute is considered to be lasting less than 2 weeks, whereas chronic can be a little bit more diffuse, longer periods, and we do worry about scar tissue formation and deformity, as well as granuloma formation. So here's histology for a granuloma. This means our body could not fight the infection successfully. It couldn't kill it. So the next best thing is to build a wall around it. So a granuloma is basically a sphere of immune cells completely surrounding an infection. So these granulomas, they can kind of grow. Some of them can appear as tumors. So this would be a benign tumor for the most part. This would be benign, but it could cause the appearance of a growth, and some people may feel that appearance of a growth is problematic. I did promise we would talk about chemotaxis and pavementing. So this is what happens when a white blood cell is traveling along happily within the cardiovascular system, like it normally does. But then there's an inflammatory reaction. So something happened, some cell damage occurred, and that's going to release chemokines. So actually cellular death, cell lysis, inherently releases chemokines. Now, just to remind you, chemokines is that word that we're sort of lumping in with cytokines. It's a chemical signal that says, hey, you guys. This is the mechanism. This is the mechanism of, hey, you guys. This is the mechanism of actually pulling those guys in. It's not that this white blood cell has a will. It's not that it's like got a little consciousness and is saying, oh, I got to go towards that chemical signal over there. It's that those chemicals being released in an inflammatory state are actually modifying proteins in the environment. So there's actually proteins lining the endothelium of this capillary bed. And in the presence of chemokines, they actually change shape and extend, and they bind to this white blood cell that's just rolling along. Think of this as an animation, it's rolling along. So those proteins bind to it, and they literally physically pull it through that vascular endothelium, and they physically pull it through into the interstitium. So this would be chemotaxis, moving along, and then pavementing is when it starts to roll. These proteins have made it sticky, they roll, and then they are pulled through. I always kind of imagine it like a nightmare scenario where you're walking along the pavement and like hands come out of the pavement and they pull you down through into like a mirror universe. So it's kind of terrifying, but it works for my brain. So whatever works for you. Okay, so inflammation, this is just another version of that visual we saw before with the blood vessel with vasodilation. It's a little bit more details though, and a combination of this and this will be on your worksheet for today, the new edition of the worksheet. So I'm going to have you go ahead and go over this as we go through it. The 3 events of inflammation leading to the 5 cardinal signs of inflammation. So I want you to go into some detail on your worksheet today about how that physically occurs, how we get from tissue damage and injury into our 5 cardinal signs. And it's kind of your choice how you address it, if you want to go up here to slide 12 and talk about that, versus if you want to incorporate these. I think I prefer incorporating these into one image to understand inflammation. So there was some kind of injury. The endothelium of that capillary bed bound to neutrophils and macrophages. There were vasoactive chemicals and chemokines and cytokines, and those all caused the emigration of white blood cells, neutrophils and macrophages into the tissue. So that leads to phagocytosis. We're back at that vasoactive and chemotactic factors. Those also helped with vasodilation, increased vascular permeability, and we already covered neutrophil emigration, so you would sort of fit that first slide into this segment here. And again, those lead to pain, heat, redness, swelling. And I did say for 5 cardinal signs, so let's go back here. 5 cardinal signs: heat, redness, tenderness, swelling, pain. So you do have to combine those concepts into one in order to successfully complete that worksheet. So inflammation is also going to lead to exudate. Now, exudate, again, this has a purpose. The purpose of inflammation is to bring nutrition. It's going to bring oxygen, and it's going to bring glucose, and it's going to bring all kinds of tissue healing nutrients that are absolutely necessary for tissue repair. And also having all of that sort of interstitial fluid now. Now remember, fluid moves through the body. When it's in the cardiovascular system, we call it plasma. When it's in the interstitium, we call it interstitial fluid. When that same fluid moves into the lymphatic system, we call it lymph. So in this case, it's just that same fluid that was released from that vasodilation during that inflammation response. So that fluid now has the capacity to simply dilute toxins and irritating substances. If there's, let's say, for example, there's a bacteria with some kind of exotoxin, right? We can dilute that with exudate. And again, we're also transporting white blood cells and antibodies through this exudate process. So exudate itself is not the problem. Inflammation can lead to problems. And those were all innate, right? Exudate is innate. Inflammation is initially innate. Bringing these white blood cells around is initially innate, especially things like macrophages and neutrophils. They work with innate immunity. That should be review for you. Whereas specific adaptive immunity, now that all these white blood cells are here, we can start participating in adaptive immunity. We can start learning how to kill the cell and developing a pathway to destroy a specific pathogen. Now, part of this relies on major histocompatibility complexes. And we never talked about MHCs before. We talked about antigens before. So remember, an antigen is a protein on a cell, usually. Mostly we're talking about cells, unless you're talking about a virus. It's a protein of the cell that marks it as self or other. So the way that I compare this, the thing I compare this to is to say it's um, like a "Hi, I'm Reeves" sticker. I've got a sticker on my shirt that says "Hi, I'm Reeves," and this is going to help me recognize every cell in my body. Every cell in my body is going to have a "Hi, I'm Reeves" sticker. Now, if a bacterium comes along and it says "Hi, I'm Bob," then my immune system knows to destroy that cell labeled Bob because it is not self. So the major histocompatibility complex is actually the gene. It's actually the DNA that codes for a variety of proteins that can then be recognized as self or other. So it's the gene for expressing specific antigens. There's a couple of different ways to consider that, but that's the way I categorize it. So antigen presentation happens because of the major histocompatibility complex. I frankly do not need to differentiate between class 1 and class 2. Immunology is not my thing, so I'm not going to have you differentiate or worry about MHC class 1 versus MHC class 2. But an immunology class would. There may be something that benefits you to learning that, but not in my course. The whole point here is that we are presenting something. We're presenting something to recognize the self as other. Now remember that when something is not recognized as self, we will create antibodies. If Bob shows up, I'm going to create anti-Bob antibodies. Now, an antibody is also going to be known as an immunoglobulin. And in general, we're going to have a sort of Y shape. There's going to be a chain, and then it's going to branch, and there's going to be antigen binding sites. Now remember, antigen is kind of the opposite of antibody. The antibody is what's floating around in plasma. The antigen is what's recognized as self or other. So an antibody is going to bind to an antigen, and it's going to help it do its things that it can do to destroy that invader. The antibody is here to help you destroy an invader. Now, you do have 5 classes of antibody, and anytime you've got 5 of something that are all really similar to each other and generally have the same purpose, we're going to focus on what makes them different from each other. So yes, you absolutely do have to learn the 5 antibody classes and their differences from each other. Starting with IgG, it's our most common type of antibody. It's the smallest, and because it's just this one single Y-shaped thing, it's going to travel during that inflammatory reaction. It's going to escape that bloodstream really easily and enter interstitial fluid. It can also, they circulate as single molecules. Just compare and contrast what I mean by circulates as single molecules as opposed to something like IgM or IgA, right? These do not circulate as single molecules. They circulate bound to other antibodies. So that's something specific to IgG, is that they do circulate as single molecules, and that size means they can permeate blood vessels and they can get into interstitium and travel easily. So this does make it the most common type. You're going to see IgG quite a bit. IgM is going to circulate as a pentamer. So penta as in 5. That's our, I think, I want to say Greek prefix for 5. So this is much larger, so it's not going to penetrate that capillary wall. It's going to stay in the intravascular pool, which means it's going to stay within the blood vessels. Now, this is going to be the first to be produced. So if you're immunized or you've got a new infection, the first thing that's going to be measurable and going to be recognizable as a lab value is IgM. Oh, they've got elevated IgM. They're in the very, very early stages of exposure. And this is going to help us activate the complement. We haven't talked about complement activation yet. These are functions of antibodies. And if you recall from AMP2, complement activation was inviting all of the white blood cells to the party. That was the getting more attention from more cytotoxic T lymphocytes and memory B lymphocytes, and bringing more and more of those elements to a site in order to better attack that invader. That's complement activation. IgA is a dimer, so di as in two. Two antibodies joined together. They're going to be found in tissues, under skin and mucous membranes. They'll be found in saliva and tears and breast milk. So I think this is the only one you're going to see in breast milk. I think that's a good way to differentiate this from others. Part of how babies get their immunity, especially in the early months, is through breast milk. Breast milk helps with the baby's immune system while that baby is still developing its immune system for the first time. IgD, again, we've got a monomer. We're going to find them primarily on B cell membranes. Remember the way we illustrated our memory B lymphocyte? It had these things that looked like antibodies sort of sticking off of it. So that would be IgD and a little bit IgM that actually are those antibodies. They're going to stimulate B cells. Their job is to stimulate B cells to do what B cells do. So remember, memory B lymphocytes are going to undergo mitosis into more memory B lymphocytes, but also, and this is where I would usually pause and see if you guys remember what they also differentiate into. They also differentiate into plasma cells. Now, plasma cells synthesize antibodies. So when those cytokines from the T helper lymphocyte invited the memory B lymphocytes to the party by saying, hey, you guys, that memory B lymphocyte differentiated into a plasma cell or a plasma B lymphocyte and secreted immunoglobulins, which, as you know, those immunoglobulins can now activate the complement and bring even more immune cells to the party. It's a positive feedback loop. It's going to keep going. We're going to keep inviting more people to this party until the pathogen is actually dead. So that was IgD. And then IgE, you can mostly associate this with allergies, also parasites. You may remember from our 5 white blood cell types, never let monkeys eat bananas, that basophils act in allergic reactions. And you also have eosinophils that work in both allergic reactions and in parasites. So you are going to see IgE also in conjunction with some specific white blood cells. I'm pretty happy just lumping this idea of IgE in with allergies. That's going to be your big payoff thing to learn. Now, it is possible for these to class switch. So we can have things change from IgM to IgD to IgG, IgE, IgA, and that will happen in the influence of specific cellular signaling pathways, cytokines, chemokines. And again, this may be something you'll see someday as a lab value if we're trying to determine if somebody has an acute or chronic condition. Now, this should be mostly review from AMP2, where we have the functions of antibodies, and we already covered activation of the complement. Again, activation of the complement is inviting more and more people to the party until the pathogen is dead. We talked about neutralization in AMP2. We said one thing that antibodies can do is they can cover the harmful thing and keep it from harming you, right? If it's a virus, it works by injecting its DNA into your cells. So if the antibody is covering that part of the virus that can inject, then it can't inject its DNA. So even though the virus is still in your system, it's no longer harmful. It's been neutralized. And the sort of dirty way of remembering that I taught you in AP2 is that it's kind of like that antibody was, it's like putting a condom on that virus. So it can't let go of its DNA. It can inject its DNA into your cells. Um, you can think of it that way if you want to, or not. It could also cover the harmful part of a toxin. It can precipitate out or agglutinate out. Now remember, agglutination is when we clump cells together. Precipitation was when we clumped harmful things together, things that were like an individual toxin. We precipitate them out of tissues or cells or fluids so that they can't land on your cells, and instead we can hopefully filter them out, pee them out, get rid of them. The one we did not cover in AMP2 is opsonization. And opsonization just means this antibody lands on the pathogen and it makes it into a flag. It acts like a flag that says this is the thing we're trying to kill right now. This right here is what needs to die. So a white blood cell will then see that this pathogen has been opsonized and it will go attack that thing first. It's going to be also easier for it to attack that thing once it's been opsonized. And again, those are general functions of antibodies. These are the tools that antibodies have to help destroy or agglutinate or remove or neutralize a pathogen. Now again, complement activation. We've been very general about what complement activation is. It's inviting a bunch of cells to the party. That complement does involve a complicated cascade. A bunch of plasma proteins are involved in inviting people to the party. I'm not going to have you learn the individual parts of the complement. I think the important vocab associated with complements, you are going to see phrasing like, again, cytokines, chemokines. You may see things like, oh my gosh, what's the word I'm looking for? No, I lost it. Again, all of that chemotaxis, all of that pavementing. Okay, so we've covered innate immunity and specific immunity. You can also sort of consider this passive versus active immunity. You can have immunity that was transferred completely passively to you. And passive immunity can be natural or artificial. Active immunity can also be natural or artificial. Here's what I mean by that. So passive immunity means antibodies were transferred, one way or the other. So passive immunity, when a mother is pregnant and there's a fetus, IgG can cross the placenta, and that's going to be part of passive immunity. This is, again, a preformed antibody. Once that baby is born, the mother passes IgA from breast milk to the infant. So these would both be natural, and they're both passive forms of immunity, which means the baby in this case doesn't need to synthesize their own antibodies. They're already pre-synthesized. Serotherapy would be the artificial version of this. Let's say there's a viral illness, and a certain percentage of people survive this viral illness, and after they survive that viral illness, they are immune to it. But some people who get this viral illness do not survive it. So for them, we may use serotherapy. Somebody's really, really sick with this virus. Their immune system is not working. It's not kicking in. So we inject them with the antibodies from the person who is immune. And now they have the antibodies in the very, very short term that'll help them destroy this invader, this pathogen, this virus. So that would be serotherapy, and that would be artificial. Active immunity is based on specific immunity, the body's own immune response. So either because we got an infection or we were immunized against that infection. So remember, second exposure means a quicker response. If you've had the immunization, then you're already going to have the specific immune response to that antigen. And I just want to clarify what an immunization is. A vaccine contains perhaps the microorganism itself, but it's going to be altered. It's going to be hobbled, so to speak. Or sometimes just the toxins, or sometimes just the antigen. There's actually different kinds of vaccines, and the more of the actual organism is in there, the stronger your specific immunity development is going to be. What I mean by that is, let's say there's a virus and we just weaken it. Now that virus, again, has antigens. It's not technically a cell, which is why I have to be careful about using the word cell. That virus has antigens that can be recognized as other. And if we have the whole virus there, then your immune system can develop very specific immunity. It knows exactly how to destroy that virus. But the trade-off here is if somebody's immunocompromised, even if this virus is hobbled, they could have a bad reaction to that virus. It's not true that an immunization can give you the disease. That's not the case. But an immunocompromised individual will be at greater risk for having a bad reaction to a strong vaccination, such as one where the microorganism is there, just weakened. So you don't want to give this one to an immunocompromised individual, but for a person with an otherwise healthy immune system, this is by far the most effective vaccine. On the other end of the spectrum would be just putting an antigen into somebody's system. Now we put an antigen into the system, we can recognize the antigen, we can mount an immune response, but we may not develop the full skill set to destroy that virus if the virus isn't there. It's just the antigen. So we can recognize the antigen, but it's not as strong of a response. So those tend to be the vaccines that require a booster shot. That's why we have things like booster shots. Because it could be a weaker form of the vaccine where our cells didn't learn exactly how to kill it. And this is a really, really big picture. This is a really non-scientific way of explaining it, but I think it works. Now remember, IgM is the first antibody to show up after an infection, so that will coincide with your first innate response. And then later into the infection, you'll see IgG, which again is the more common one, the one that you have with the highest frequency. On your second response, it's going to take less time to respond very strongly to a pathogen. So there's a less time between innate and specific responses. And you won't need as much of your innate response because that specific response is going to kick its butt really quickly. So that's what immunity looks like. And again, these may be something you see as lab values. I'm going to go ahead and stop there for our first hour. It's only been 38 minutes. And then I'll start again with alterations in immune function.